Hello, today is March 22nd, 2018. My name is Jeff Sands, and I am at Emory University School of Medicine to interview Dr. Douglas Eaton for the American Physiology Society's Living History Project. Dr. Eaton has been an APS member since 1981, and he has been affiliated with Emory University School of Medicine since 1986. Doug served as the Society's president in 2005. While Doug's research has seemed to vary over his career, in actuality, the central theme has been how membrane ion channels are regulated to produce cellular and organismal electrolyte homeostasis. Doug, welcome to the Living History Project, and thank you for agreeing to be interviewed today.
Well, it's a great honor to be associated with this Living History Project. I do remember when I was sort of scientifically growing up, that one of the wonderful things that always happened whenever any of the senior folks deigned to have us in their presence was that they would tell stories about their colleagues and their mentors and everything. And so it was one of the better, really wonderful things about that period of time. And so in a way, we're kind of doing the same thing. It's not quite as informal, but we'll try to keep it as informal as possible. Now, I should point out one thing. My interviewer here, Dr. Sands, is about to be the new president of the society come this April at the EB meeting. So there seems some real continuity in this process. So someday I'll interview him.
So in some ways, we're having a summit conference.
Right.
Past president to future president, and it's an honor to get to do this. I do have a few questions to ask you. Can you tell us where you grew up and provide us some background on you and your family?
Well, my dad worked for a defense contractor, so that meant every 4 or 5 years we would move. So I grew up all over the place. I was born in South Dakota, but I only stayed there for 6 weeks. This was right at the end of World War II, and so my dad was stationed in different places. He had met my mother because he was a radio operator, and it turns out that central Wisconsin, where she comes from, is a great place to send long-wave radios all over the world. And so they had a 100-mile radio array. And the story goes that he used to love, radios were rationed during the war, and he used to love to take old broken radios, a bunch of them, and put the pieces together to get one functional radio. And when the OB that delivered me, when my dad asked him what it was going to cost, the guy says, "It's not going to cost you anything but a radio." So I guess that's what I'm worth. And so we lived in many places. We were associated, my father was associated with the beginnings of the manned spacecraft program in Florida. And then we moved, I was a teenager in Philadelphia, went to school there. So we lived in many different places as I was growing up. My best friends were always my brothers because they were always with us, and that was nice. We still have a very close relationship.
Yes, now I understand your passion for rebuilding computers from spare parts.
You got it.
Can you tell us a little about what got you interested in science?
You know, I cannot remember a time when I wasn't interested in some aspect of natural history. Some of my absolute first memories were looking at multicolored beans in pea pods. In retrospect, I felt a little like Mendel or something like that. So I always knew that I liked science. I always knew I wanted to be a scientist. It was only a matter of sort of narrowing down exactly where I wanted to go. And I thought by the time I got to high school, I thought I was going to be a chemist. And that's sort of how I decided what colleges to apply to.
Well, I know you got your undergraduate degree in biology from the California Institute of Technology. So what led you to pick Caltech for your undergraduate institution?
Well, as I said, I knew I wanted to go to some school that was strong in chemistry and technology. And I had been admitted to both MIT and to Caltech. And we lived in Philadelphia. So my parents were pushing me to go to MIT. But at that time, the tuition at Caltech was one round-trip airfare less than the tuition at MIT. And I thought going out to the Great West would be a wonderful thing to do. And as it turned out, it was. Now, the fact that I was a biologist was sort of an accident. I was a chemistry major until the second quarter of my senior year. So in chemistry at Caltech, you had to have 2 years of a foreign language. And I'd taken 2 years of French already. But in the second quarter of my senior year, my advisor said that he thought that anybody that graduated from Caltech ought to have a full year of German. Well, I wasn't too interested in sticking around for another year to fulfill that requirement. So that afternoon, I became a biology major with a chemistry minor instead of a chemistry major with a biology minor. And the rest is history after that.
Well, and very fortuitous for physiology that it worked out that way. After graduating with your Bachelor of Science, you decided to pursue a graduate degree and went to the Scripps Institute of Oceanography as part of University of California, San Diego. Can you tell us why you decided to pursue a master's degree in marine biology and share the names of the people you worked with at Scripps?
Well, that's another interesting story that I was interested in neurophysiology. That came about because of a course that I took with Felix when I was a senior at Caltech, where we were introduced to neurophysiology. And I decided that I wanted to go to graduate school in that area. And in fact, in the area of cellular neurophysiology, single-cell neurophysiology. Mammalian preparations were not accessible at that time. And so you worked on some sort of invertebrate. You either worked on sea critters or you worked on bugs. We used to call ourselves the squishies and the crunchies. I was a squishy. So everybody that was a neurophysiologist that worked on squishies worked out of marine labs. Well, Scripps was the closest marine lab, and they were trying to establish a neuroscience program there. It would have been the first in the country. It did turn out to be the first in the country. And so there was a real strong component of neurophysiologists there. Ted Bullock, Galambos, Livingston, all the sort of big names in neuroscience at that time were present at Scripps. Now, Scripps pulled a trick on me. I would have graduated with a PhD in marine biology. I mean, you were at Scripps, you were some sort of marine person. They also changed their language requirements when I was starting my thesis work. And I said, no, I don't think so. And the neuroscience program, the graduate program, had opened up the week before. And so I petitioned for a master's degree in marine biology and then became the first of 2 graduate students in the neuroscience program there. So so that's the way I became a neuroscientist. It's silly the way that you sometimes make choices, but it worked out.
And as you mentioned, you then remained at UCSD to obtain your PhD in neuroscience. Can you tell us who you worked with and what your thesis project was on?
I was actually working with Susumu Hagawara. He he probably doesn't get enough credit for it, but he discovered calcium channels and calcium excitability in excitable tissues. And he'd talked about it for years, and then went on. Other people picked it up. Some Dick Chen at Yale and so forth picked it up. And so he was a wonderful mentor to learn about electrophysiology and biophysics. He sadly died kind of early. He was in Japan during the war and got tuberculosis, and the treatment for tuberculosis at that time was poor, and so there was always some stress on his heart, and finally it got to him. The other people, as I said, I was the first of two graduates in the neuroscience program. First neuroscience program in the country, first of two neuroscience students. So he was kind of proud to say, he always pointed out that I was his first American PhD student. He'd had some Japanese students prior to that time.
And after receiving your PhD, you moved north up the I-5 and I-405 to LA to postdoc at UCLA with Susumu Higawara and Jared Diamond. Can you tell us about the research you pursued and what it was like working with these two internationally recognized scientists?
Well, I've already told you about Higawara. So we did some additional work on calcium channels. If you look way back in my CV, there's some articles on that. The way I got to work with Jared was a little strange. That he had a graduate student, Simon Lewis. You probably know Simon. And so Simon worked down the hall from me, and one night he was really down, and I said, "Simon, what's the matter?" And he says, "Oh, I'm going to be out of date before I get my degree. The folks in Germany have already started to use microelectrodes on epithelial tissue, and I don't even know how to do that. And I'll be out of date, and I'll be an old fuddy-duddy before I even graduate. I'll never get a job." And I said, "Well, wait a minute, Simon. I mean, that's what I do. Next time you have a preparation, I'll just bring it down to my lab. Let's take a look at it." And we did, and we were actually got the first publication on looking at microelectrodes in epithelial cells, bladder cells. And his mentor was Jared Diamond, and Jared said, "Well, I know you're a little bit at loose ends here. Why don't you start working with us, and we will do this work on epithelial tissue?" And I said, "Oh, okay, that's great." And Jared was a wonderful guy to work with when he was in town, not in New Guinea looking at birds or something like that. That was his other love besides the physiology. So that's the way we worked together.
You also did a postdoc with Felix Strumwasser at Caltech. Can you tell us about that experience, as well as about your experience as a participant of the second Alpha Helix expedition to the Great Barrier Reef?
Well, of course, they were quite separate from one another. Felix ran a laboratory where he was doing electrophysiology on circadian rhythms in gastropods. Again, this issue of he was a squishy also, and so he knew that I was interested in that, and so he was going to go on sabbatical, and he says, "Why don't you come over here and take care of my lab while I'm gone? I know that pretty soon you're going to move to your first faculty position in Texas, so it'd be a good interlude." And the beauty of it was it gave me the opportunity to actually continue to work with Simon Lewis and Jared Diamond and to do some work with circadian rhythms in the gastropod neurons. Now, the Barrier Reef expedition was out of Scripps. Scripps had this research vessel that was designed to do physiology, actually. And again, because we were squishies, a research vessel that could go all over the world and find the best squishies around was a wonderful opportunity for us. So we were actually interested in the fact that certain classes of fish, like pufferfish, we wondered why they were not susceptible to the toxin they made. They make this toxin that wipes out nerve cells. That's what kills people when they eat when their pufferfish is not prepared properly. But it doesn't kill the fish. And it turns out that they have a variant of sodium channels in their nervous system, the excitable channels, that is not susceptible to tetrodotoxin. So we wanted to look and see how far that went, and it turns out it's not just the pufferfish. There's a whole order of fishes that are not susceptible to tetrodotoxin. So that's what we did, and again, if you look back far enough in my CV, you can find an article about that.
In 1973, you joined the faculty at the University of Texas Medical Branch in Galveston and also became investigator at Woods Hole Marine Biological Laboratory and Marine Biomedical Institute in Galveston. Please tell us about your experiences establishing a new laboratory and about your research program.
The people that went to that department, the department had sort of fallen by the wayside, and so the university was going to try to reestablish the entire department. So they hired Arthur Brown, Buzz Brown, to be the chair, and he was allowed to hire 9 additional faculty to come in and reestablish the department. And they didn't spare any money. I mean, what it amounted to is that we were bought, you know. And so that meant establishing the new laboratory was easy. You know, we had plenty of money, and we had plenty of money for the department and plenty of money for individual laboratories. You made up a list of all these things, and you were a little embarrassed about it, but they didn't blink an eye, and they wanted to know when we were coming. So that was simple. But at that same time, there was a fellow there by the name of Harvey Fishman who had worked with Casey Cole and worked with some other investigators on squid. And he said, "Oh, you boys," he was older and we were younger, "you boys come along with me to Woods Hole and let's do squid axon together." And so we did that for 15 years, I guess, before we did that. Now the Marine Biomedical Institute was interesting, is that they wanted to use marine animals for electrophysiology and neurophysiology also. We had this idea that we were going to make Galveston, at where University of Texas was there, Woods Hole South, because the same squid that were up by Woods Hole in the wintertime were down by, or rather in the springtime, were down by Galveston in the wintertime. The bad news was that they were 200 miles offshore, so they were there, but we just couldn't get them very easily.
In 1986, you joined the faculty here at Emory University School of Medicine and became deputy chair and then chair of the Department of Physiology in 2008. Can you tell us about your department, the people on faculty, and the role the chair plays in shaping the department and its direction?
Well, of course, the chair, when I came, the person who recruited me was Robert Gunn, who also was a very strong member of the American Physiological Society. Unfortunately, he contracted or came down with lymphoma and died in 2005. And so the department had two areas of strength: membrane transport and cellular physiology, and neurophysiology. And then I sort of bridged the two areas there between those two. And that was when we really started to be involved in renal physiology as well. And of course, that's how I happened to have met you, who came shortly thereafter and really became involved in the division of nephrology at Emory. So we had a real collection. It was a very strong department. We lived in the basement. That was good news and bad news. It was bad news because we found out for sure that water rolled downhill. It was good news because nobody else wanted to be down there, so we had the whole basement to ourselves and all the facilities that were associated with the basement. And if anybody ever showed up down there, you knew that they must be serious. They were either lost or they wanted to be serious about talking to you about anything. So it was a wonderful time. After Bob died, they started an open search for chairman, and it did not go particularly well. And it got to the point where finally the then dean, Thomas Lawley, said, "Well, why am I going to all this trouble? We have a pretty well-known physiologist here. Let's just make him chair." And that's what happened. Matter of fact, just the day before yesterday, I was cleaning out some files and I found a letter from him that said, "Oh, welcome on board," and so forth. So we had two goals. We collectively, being the faculty that were there and me, one, to develop neurophysiology more. We already had strength in that area, and then to develop membrane transport. We did a great job of developing the neurophysiology. We recruited quite a few people.
Okay, start that question again.
Okay. Doug, the central theme of your research has been how membrane ion channels are regulated to produce cellular and organismal electrolyte homeostasis. However, as you've mentioned, early in your career you worked in nerve cells, while later in your career you worked in epithelial cells. Can you tell us how these areas are related and why you made the transition?
Both cell types require their own cellular homeostasis. So that's sort of how they're related. Now, when I started out working on nerve cells, they were the only cells that people thought ion channels were important, that they were important to maintain the excitability of the nerve cells. That's what nerve cells did. And most folks didn't even imagine that epithelial cells had ion channels in them. And I kind of thought that couldn't possibly be true, that if they transported ions, they had to have ion channels. And so that's when we began to measure the electrical properties of epithelial tissue, and it turned out to be pretty fascinating. And we were one of the, if not the first, one of the first groups ever to look at ion channels in epithelial cells transporting sodium. So that turned out to be more fascinating than I'd ever thought. And I still dabble. I mean, I think 2 years ago I had a paper on a certain type of potassium channel in nerve cells, but it's just dabbling. That's that's hobby science when I do neuroscience now. The real work is in renal physiology or well, not just renal physiology, also pulmonary or lung physiology too.
The fact that you worked in both excitable cells and epithelial cells is the reason why you were recruited to serve on the physiology study section. Tell us about your experience on the study section and about the importance of participating in such endeavors.
Well, first, the peer review system is fundamental to the way that we do science in America. I mean, there may be issues with the peer review system about the way that things get reviewed, but it's far better than any other system that you could have. People have talked about having sort of use the Chinese system or the Japanese system or the German system, where the big boss gets a lot of money and then he distributes it out to sort of junior faculty around him. That's never been in our culture, and I think the fact that U.S. science has been so successful means that that system works. Okay, but it only works if you have people who are willing to do the reviewing, and it's not a trivial task to ask people to do. But you know, when I was asked to go on the physiology study section, that was a problem. At that time, grants were on paper and they could be as big as you wanted, and you'd get this enormous big box in the mail filled with all these grants that you had to review. And I remember my kids were kind of small at that time, but they hated seeing that box come in the mail because they knew Dad would be grouchy for the next few weeks while he was going over that. So it was an important time. And you learned an enormous amount by being on the study section. I mean, you have the same experience, so you know exactly what's involved in that. And because obviously the physiology study section was a love of mine, physiology after all, but then because of the work that I was doing with kidney and so forth, I also served on the kidney section of general medicine B. So several different study sections, and they're the underpinnings of U.S. science and the NIH granting support to scientists in the country.
As you say, I had the privilege of serving on General Medicine B while you were the chair, for at least for part of that term. And I think my kids would agree with your kids in terms of the pain of seeing that big box arrive. Doug, I noticed in your biography that you direct the first program that pairs Emory University with 3 minority-serving institutions in Atlanta: Spelman College, Morehouse College, and Clark Atlanta University. Can you tell us about the program and your commitment to enhancing diversity in the sciences?
Well, this program was an experiment to start with. We thought that there might be if not a better, certainly a different way for postdoctoral, to educate postdoctoral fellows. That we thought that in this day and age, not only should they have a really fundamental research background, that's what every postdoc is supposed to have, but they should have some experience with teaching and presentation, because we thought that no matter what they were going to do, that they were going to be called on to either teach or present, do things that involved communication. There were some subsidiary goals also, one of which was by having this partnership with the HBCUs, the historically black colleges and universities, the 3 that we're partners with, that our fellows would be exposed to a diverse environment, so they would have experience in teaching in that environment. And the other benefit of it is probably because we're in Atlanta, because we're partnered with the Atlanta University Center schools, that we recruit some of the best, most well-qualified minority postdocs in the country. About half of the program are minority fellows. And that means that we place minority fellows in academic positions all over the country. We have an enormous network now. 184 postdocs have graduated from the program over its 18 years, and we have them everywhere. If you had a map of the country and you had dots on it, it would look like would look like the United States had measles or something like that. So we're quite proud of that program, and we're actually every year there are more than just our program. There are 22 other programs like it now. We started out with 3. There are 22 other programs because it's been so successful. This idea has been so successful. And we have an annual meeting where we all get together and we discuss issues associated with career development and with development of the programs. And the meeting's going to be here in Atlanta in July of this year. So we're out trying to raise money for it. If you know any wealthy donors, let me know.
So what do you think has been your most significant contribution to physiology?
I think there are probably sort of 2 areas. One area, as I say, I started working on epithelial transport where at a time when everybody thought that epithelial cells were magic. I mean, magic in the way that that the rules that applied to nerve cells and so forth didn't apply to epithelial cells. And that by using the methods that we use, the electrophysiology and chemical methods and biological methods, we could sort of dissect individual cells and show that in every respect they behaved just exactly like you would expect other cells to behave, except that they had the 2 ends. They had the end that faced the world and the end that faced the inside of the body. And so everything about those was just the way it was in other cells. They had ion channels, they had transporters, everything was the same. So that made some people mad, and it made a lot of people happy, so that was good. The other thing was actually about the ion channels themselves. As I said, we started to use this method called single channel patch clamp, and that's where you take a little electrode and you push it up against the side of the cell, and because of the charges on the glass and because of the charges on the cell, they seal together. And if you're lucky, you get 1 channel protein inside this little patch of membrane that you've isolated. And I joke with my friends that that's real molecular biology, 1 molecule at a time. And the beauty of it is that you can actually pull that little patch of membrane away with the ion channel and get at the inside surface of the cell, and that's where all the regulation takes place. So I think we made contributions to understanding how ion channel regulation takes place, particularly in these epithelial cells, like in renal epithelial cells, and that has real significant consequences for how the kidney and the lungs work. The kidney to control blood pressure, and the lungs to control fluid balance, and particularly it has consequences for pathology. You know, what happens when you get pneumonia? What happens when you have high blood pressure? Why do you have high blood pressure? Things like that. I'd like to think that that's our contribution.
So tell us about your first APS meeting and how it differs from current scientific meetings.
APS used to have what they called a fall meeting. It was a small sort of specialty meeting. It was not like the big experimental biology meeting or the Federation meeting is what it was called at the time. So my first APS meeting was one of these little fall conferences that actually had to do with neurophysiology. It was ion channels and neurophysiology, and it was at College Station up in Pennsylvania, at Penn State University. And that was still at a time, where it wasn't trivial to go from the West Coast, where I was, back to the East Coast. So this was sort of a big deal to us, and it was the first scientific presentation that I made. And I met a whole bunch of folks. Part of the reason I was going was that Felix Strumbosser, who I had been working with at that time, had just received the Bowditch Award. And the Bowditch Award, the central award for the APS, was usually given at these fall meetings, not at the spring meetings. So he was there, and so I was going along to lend moral support and to make my first presentation. And it was a wonderful networking opportunity, because there were all these other young investigators, most of whom I hadn't known, who were from the East Coast. You know, we were the West Coast group. Margie Anderson was one person who's still up at the University of Massachusetts, and there were some other people who still we connect with one another even after all these years. I mean, what was that, almost 40 years ago, I guess.
So Doug, you've been an active member of APS, serving as its 78th president. Can you tell us about the role that APS has played in your career?
APS has actually been central to my, what I would think of as my adult scientific life. I joined APS kind of late. It was hard to be a member earlier on, so I joined in '81, and ever since then, it's played a really significant role, and I tried to contribute to the APS. I was initially associated with a cell section, doing all sorts of programming for them, and then I sat on their executive committee, and I was finally the executive or director. I've even forgotten what it was called in those days. And the other thing, I was involved in diversity affairs. I was on the Porter Committee. That was the first committee that I served on, and I still remember some of the fellows and the people that served on the committee with me at that time, Pamela Gunter Smith and others, who's now a president of an HBCU. So that's great. And it always provided a venue. The meeting, the EB meeting, is an enormous meeting, but it always provided a venue where you could present your stuff and you could really interact. Everybody talks about the importance of networking, and you know, that gets a little hackneyed after a while, but it's true. Why do you go to the meeting? Well, you go to the meeting to hear the latest science and to meet your scientific friends. And you don't oftentimes get a chance to see them other than that. So learning new things is the day that I can't learn new things. I go to the meeting, and I think that if I come away from the meeting with one new good idea, then I haven't wasted my time. And usually it's a lot more than one, but that's the important part.
I know you've been involved in training students at all levels, from undergraduates, medical students, graduate students. What advice would you give to students today?
Be flexible. I mean, it's a more difficult, I think. I think I probably had it easier scientifically growing up than students do now. But keep your options open. so that you have, you know, so that you're willing to do something else or go someplace else. I remember talking to one of my students that said, oh, well, he was going to go to Florida. And I said, boy, that's kind of boxing yourself into a particular thing. He says, well, I want to go to Florida. He finally got to Florida, I must say. It was persistent, but it wasn't very flexible. So I think you have to, as I say, keep your options open. It helps to learn lots. I'd like to think that I have some information to impart on occasion. I mean, not just scientific, but maybe some ways to deal with different issues. So it's been great. It's been great. I mean, I've had a lot of postdocs, a few medical students, not so many graduate students, but they've all been wonderful people, and I still, I now have, as some of my former postdocs like to say, I now have scientific grandchildren because they put their students onto faculties around the country too. So it's a big joke. I'm not waiting for the day when I become a scientific great-grandfather.
Well, Doug, well, thank you very much for participating in the APS Living History program. I've really enjoyed this opportunity to talk with you, to learn more about you and your career, and I really value the time, not only this hour, but the fact that we've worked together for almost 30 years now has been a wonderful addition to my career, and I'm sure those who watch this living history video will benefit from your sage advice much as I have done over the past 30 years.
Well, you know what they say about advice, that advice is cheap and you get exactly what you pay for. So I hope that it is worth something. And Jeff, the idea that you are the interviewer for me is really special. Thank you very much.
Thank you.